So one of the things that we want to make about cholesterols, or at least some notes that we could convey about them, is that they all have, in terms of their structure, the only thing that I think, well, I mean, knowing the entire structure of a cholesterol is good, but they all have, at the basis of their structure is known as a nucleus. So the nucleus of our steroid, at the base, something that does not change, is this polycyclic ring. Hopefully you can see that. I don't know if this is probably not the best drawing. You can find on Wikipedia a lot of better drawings. But it's a polycyclic ring consisting of 5 total rings to it, 2 of which are 6-membered and 1 of which is, not 5, sorry, 4-membered rings, 2 of which are, or 3 of which are 6-membered, 1 of which is a 5-membered ring. And in the context of a cholesterol, what we will have attached to it, and I'm going to draw it in like this because it's a variable region, is an OH group, some type of a polar in there. And if I wanted to make this an a steroid, one of the more common ones here, I'll draw a double bond there. And then at the very edge of this, I will have some type of a hydrocarbon R group that tends to variate with each structure. But there's lots of different ways that we can make cholesterols. Estrogen, testosterone, those are both just cholesterols. I think for my organic chemistry final, I was like, I had to synthesize, I think it was estradiol from a petroleum. That was fucking insane. But anyways, this is how this stuff works here. So what observations can we be made about the structure of this? Well, structurally speaking here, we can say that it is, I'll switch to blue, amphipathic, right? So it's both polar and nonpolar. If I were to draw this out here, I'd say that it has a polar property of the OH group there, and then the nonpolar would be pretty much the rest of the molecule. But in this context, I'll say it's nonpolar is all this jazz over here. I'm getting kind of bad at color coordinating this stuff, but who cares? So for the function of cholesterol that they have here, there's really just 2 important functions that they have. There's one that helps with membrane stability, and then the other one that's acting as steroid hormones. All right, so for the concept of membrane stability, we have things that are going to keep us stable whenever it's hot outside and things that are going to keep us stable whenever it is cold outside. All right, so the one misconception that a lot of people have is that plants don't make cholesterol. They do, okay? They don't make a lot of it. Okay, don't go, go crazy. They do make cholesterol. They don't make a lot of it, and they sure as shit don't use it to the extent that we use it, but they do have cholesterol in their membrane. 20% of the bacterial species that we are aware of have similar like structures to cholesterol. This is a universal thing. This is not something that is limited to animal cells. Okay, if 20% of the bacteria that we've been able to observe can formulate cholesterols in the form of hopanoids, which are cholesterol-like molecules, I guarantee you something as complex as well in terms of natural selections as plants, they do have cholesterol. Okay, I know that, and when we were talking about in general biology, we did a very broad overview of things, and we didn't get down into the nitty-gritty and the specificities of stuff. And generally speaking, we say that they don't have cholesterol. That's true, generally speaking, but they do make cholesterol. It's driving that point home. But anyways, so let's say, for example, if it's hot outside, and if it's hot outside, our membranes start moving around pretty frequently. So if we're moving around pretty frequently, the way that I look at this is because of the structure here, because of this amphipathic nature of it, it's going to be able to form bonds with the outside and the inside. And so it's going to act kind of like a sandwich between the phospholipids. I spelled that wrong. I'm sorry. But anyways, if you could imagine, you have yourself a cholesterol here. I'm just going to draw it like that, but this is what it would look like in the membrane. On either side of it, we have a couple of fatty acid phospholipid molecules here. It's getting kind of hard to draw this. It's late. I'm tired. And there's another cholesterol molecule there. Now, unlike these guys, well, these guys do have other connections, but because the cholesterol can be linked strongly to the outside or linked strongly to the inside, they don't really tend to move so much. They can sometimes clump together and form lipid rafts, which are freaking cool. Talk to Kai Simmons if you want to know about that stuff. But if there's only a finite amount of room, so say if we increase the temperature and these guys start moving around really, really fast, which I'm shaking with my pen to show that they're moving fast, they're not going to just fly off and form a pore, right? Even in extremely hot temperatures, they're just not going to form a pore because these cholesterols are acting in that format. By the same notion, though, if it's really cold outside, cholesterol is a physical spacer. It's going to physically have space between these phospholipids and then these phospholipids on the other side, right? So there's going to have some degree of permeability. They can't all just come compacted closely together in the format. I guess if it's cold, I should have drawn them as saturated or kinks in their dough from the cis bonds that are formed in there. But anyways, that's what's happening there. Now for steroid hormones, what's interesting about steroid hormones is that because they're just that, they're steroids, then they're going to have intracellular effects. Okay, so they're fat-soluble hormones if you think about it this way. And therefore they're going to have really intranuclear receptors. And the consequences of this for steroid hormones is these have really long-term effects. So to give you an example of this, if you've ever experienced a fight or flight response, the main predominant molecule of that is epinephrine, but you're not in a state physiologically of an accelerated heart rate, and you're not in a state physiologically of being alert and physioactive. That state doesn't last for a long period of time. But what does have long-term effects would be the cortisol effects of it. So an example would be cortisol. That's a stress hormone. Decadron is the clinical term for it, but we call these glucocorticoids because they're kind of corticoid sterol-based hormones. Testosterone, sex hormones would be an example of this. And so these have long-term effects because they're not lipids, because they are lipids and they're not binding in the format of a protein. Proteins are very easily, they bind on the outside of the cell and they get slowly drifted away. But once these things get inside our cells, they have very long-term effects because that's the way that works. Comparing, I guess, just to really drive this home point, so compare cortisol with insulin. Okay, insulin does his job really quickly. Insulin's a peptide hormone. Cortisol does not do his job very quickly. Cortisol is a steroid hormone. I feel like I'm baffling on too much about that, but that's how those work.